Okay, can I get everyone's attention, please? I think we can get started. Okay, thank you. So it is a real pleasure to have Steve Quake visiting us to give this lecture both today and yesterday. It's been wonderful to meet you and to learn more about your work. So just briefly to introduce myself, I'm Sarah Rocher. I'm a faculty member in computational biophysics, and my lab is located at the University of Toronto, Mississauga. And I'm really pleased to be introducing Steve Quake. He is the Lee Otterson Professor of Bioengineering, and he's also a Professor of Applied Physics at Stanford University. He was the founding co-president of the Chan Zuckerberg Biohub. He received his bachelor's degree in physics and a master's degree in mathematics from Stanford University. He then went on to Oxford University to complete his PhD in physics. Following his doctoral work, he moved to work with Stephen Chu in single molecule biophysics for his postdoctoral research. And then he joined the faculty at Caltech. Then in 2005, he returned to Stanford University, where he helped to launch a new department, the Department of Bioengineering. He has made tremendous advances at the interface of physics and biology. He has been using quantitative tools to illuminate what some might think of as the dark matter of biology. So relating a little bit to the earlier talk today. His research program has shaped multiple fields, including single molecule biophysics, DNA sequencing, and microfluidic large-scale integration. His work has been both fundamental as well as consequential for human health. His inventions have led to transformative clinical diagnostics, including the first non-invasive prenatal tests for aneuploidies. And importantly, several companies have also grown from this work to actually bring these technologies into clinical practice. I read an interview recently with Steve by the Caltech Heritage Project, and in that interview, he quoted Louis Pasteur, who said, "There does not exist a category of science to which one can give the name applied sciences. There are sciences and the application of science bound together as the fruit of the tree which bears it." And I think that this connection between science and applied science is really evident in Steve's body of work. His work has actually clearly shown that the boundary between physics and biology is actually one of the most productive places where a scientist can stand. For all of these many research achievements, he's received numerous awards and has also been elected to several societies. His awards include the MIT Lemelson Prize, and he is an elected fellow of the American Academy of Arts and Sciences, the National Academy of Inventors, the National Academy of Sciences, and the National Academy of Engineering. And most recently, he was appointed as a foreign member of the Royal Society. Please join me in welcoming Stephen Quake.
Thank you so much. And thank you all for sticking around for the 4th hour of physics the past 2 days. And you guys are the true diehards. I'm going to pick up where I left off yesterday on a couple of topics and get into some more recent work and things that are more, let's call it physics oriented aspects of it. And then maybe talk about something a little older. So remember we were talking yesterday about how the genome is not the blueprint of the organism. There's no way to predict the cell types just from the genome alone. So we went and spent the last decade measuring that using single cell transcriptomics in a variety of organisms, mouse, fly, lemur, human. In human in particular, we worked very hard to get multiple tissues from the same donors, and this created what we call the tabula sapiens. Someone was asking me in the question section yesterday about of aging and such forth. So I thought I'd start off with that. We'll start off with a little biological vignette about aging in the tabula sapiens. And there's something called the Hayflick limit. Hayflick was working at a time when people thought cells were immortal. Primary cells could just divide and divide over and over again. And the biggest luminaries of the field, very famous people, had published papers claiming that's the case. And Hayflick actually found the opposite. That wasn't true. Primary cells are not immortal. They'd only divide around 50 times before stopping. And this was a big challenge to the dogma and the orthodoxy of the day. He struggled to get it accepted and published. He was right. The other experiments suffered from issues from contamination and such forth. But he eventually sort of prevailed. And you can sort of calculate from this notion based on sort of how many cells we've gotten, starting from one cell. Maximum predicted human lifespan is about 120 years, theoretically. And that's more or less what's observed. When the cells stopped dividing, they entered a state that he called senescence. He sort of coined that term. And the study of senescence has become central to understanding aging. There's been a ton of great sort of discoveries about the molecular basis of senescence over the years. Hayflick himself passed away 2 years ago, and someone at the New York Times had great fun writing his obituary headline, which is Leonard Hayflick, who discovered why no one lives forever, dies himself at age 96. He made it to a ripe old age. So we decided to look at senescent cells in the tabula sapiens. And for those who are experts, we're looking at cells that are CDKN2A positive, MKI67 negative. Looked through the atlas, found something like 50,000 of them in the atlas. And they're there at about a couple percent level, a few percent level, which is sort of what you'd expect. And this turns out to be the largest set of naturally occurring human senescent cells ever pulled together in one place. So a very interesting resource to try to understand something about how senescence works. And my postdoc, Madhav Bantry, did some really terrific work here doing that. And the first question he asked was, are all senescent cells senescent in the same way? In other words, do they share a common gene expression program other than expressing CDKN2A and not expressing MKI67? And so the answer to that is no. There's not a common expression for senescence. They're not all the same. And then if you look at differential gene expression of senescent cells of a certain type and non-senescent cells of that type, and ask what genes are common across cell types, there are a few that show up as shared between some, but not all. Some of them known, some of them novel. But the overlap is not as high as one might expect. And so this has implications for people trying to develop anti-senolytic drugs that'll slow down aging. It's only going to work in certain tissues and cell types for any given drug. You're going to have to think about a more sophisticated strategy to address these different kinds of senescence in different tissues. But all is not lost. The senescent cell types are not completely each one unique. There are sort of broad programs being used, and we use non-negative matrix factorization to kind of work that out. There's about a dozen to 16 broad gene expression programs that are mixed and matched across the different types of senescence. And you can see here, one way of looking at the data is to see them separate into 6 broad classes. This is according to cell type and not tissue specific. The top is colored by cell type, the bottom colored by tissue. And you can see that these are cell types that are shared across tissues, yet still share similar senescence programs. And so we're continuing to dig away at that. And it's been a very, very fruitful avenue and an amazing data set. There's going to be much more to come on that in the next year or so as Madhav continues his work. Now I'm going to turn to a different problem, which is how we determine the cell type, the cell names, the function of the different cell types. And this we've done, I said yesterday, using expert annotation by biologists who study those cell types. But we got interested in the question of, are there other ways to do that that don't require biological priors and are sort of principled? And a terrific student in my group, George Crowley, has been working on this for a while. His paper was just published a month or 2 ago. And it came out of a sabbatical visit by Uri Alan, who had been interested in, for years, more than a decade ago, trying to use the ideas of Pareto optimality to understand evolutionary trade-offs in biology more broadly. And we, after hanging around the lab, decided let's try to apply this approach to the entire tabula sapiens. So the challenge has been, how do you do a first principles definition of cell type? That's been hard. We want to do it in a way that's principled, unbiased, and identifies the functions performed by each cell type, ideally without requiring any prior biological knowledge. That's, physicists, this would be great. What if we kind of short circuit all of biology and we just cut to the chase somehow? And Pareto optimality is a very clever mathematical approach, came out of economics, end of the 19th century, which describes how if you have a problem with multiple objectives, how trade-offs between them will constrain the set of optimal solutions. Let me just say a word about that. So the definition of a Pareto optimal state is a state where no ability can be improved upon without making at least one other ability worse off. And you can see that here. If there's two tasks you're trying to perform, task one is the x-axis, task two is the y-axis, and sort of perfect performance is on the axis. That point right there, V prime, is on the front where you can't move in any direction without making one worse off. Whereas if you were in the interior, you could move in a direction and improve one without making the other worse off. So the solutions tend to be on the boundary on a front. And if you move from task space to trait space, and the traits are the things that allow you to do tasks, or in this case, the traits are the genes, you can see how the geometry sort of works, that the two limits on the axes are the archetypes, 1 and 2. Sorry, is there a pointer here? No? Well, I'll just wave my hands. So this is the one archetype. The other one is on the other axis. Those are the 1 and the 2 there. And if you believe that, if you assume that performance falls off, say linearly with distance, thank you, Tony. All right, terrific. If you believe performance falls off linearly, you can show pretty quickly that that front must be on a straight line between these 2 points. Because if you're off the line, you can improve both of them by moving towards it and shortening the distance to the archetypes. And so the front becomes a straight line when you're moved from task space to trait space. And Uri went and showed in this paper 14 years ago several really cool examples of that, using not molecular properties, but macroscopic properties of biological systems. And this is like ratio of tooth size ratio of different molars in different creatures that eat plants or meat or both. And you can see that sure enough, these ratios for different species lie more or less on a line. And one set of species is more optimized for this task, other ones for this one, and it kind of works. And this generalizes, if there's more than 2 tasks, you go just to higher dimensions. And what you should get, same kind of argument, is a polytope. So you get, if you could keep your hand down, that'd be great. Triangle in 3 dimensions, or you can get 4 tasks, you can see that you get a sort of pyramid. And this also is something that Uri explored in another example here. This is comparing mass to longevity, and you can see that this was 3 different tasks being optimized, and the data falls within a triangle across a huge number of species. So we decided to apply this to our gene expression data for the different cell types. And George went through and did this for every cell type in the Tabula sapiens. And sure enough, they all sit in polytopes. Some of them triangles, some of them pyramids, but some of them a little higher dimensional. But it really was remarkable that virtually every cell type we looked at satisfied this test that falls within some kind of polytope. And if you look at the p-values of the fits, some of the distribution is something like this. Okay, so just almost all of them are fit. There's a few that are not significant, but the majority statistically satisfy that criterion, which is terrific. And so that's one important part of Pareto optimality, is to show that that's satisfied. The other part is to show that there are at the vertices enriched skills or traits. So we went through and we started to look at the vertices of the different triangles and pyramids and for different cell types, and asked which genes have their expression enriched at the vertices relative to other parts of the polytope. And sure enough, very sensible biological results emerged. So you can see here for macrophages, which are cells of the immune system, across many different tissues. They're found in many different tissues. You can see they're specializing in genes. This is the genes that are enriched at one vertex. And these are genes related to antigen presentation. and you can see that their differential expression here is higher. At another vertex, it's macrophage activation and immune response, which is the definition of the cell type when you use biology. But it emerges here hypothesis-free. Inflammatory response, they're involved in inflammation in the immune system, and another inflammatory set of pathways here. So it all kind of makes sense. Similarly, same thing for natural killer cells. These are T cells also from the immune system, and the things that emerge from their vertices are things around cytoskeletal organization, which is how they move around, inflammatory, cytokine T cell activation, that's how we define it biologically, innate immune response, that's the system they're part of. And it all, again, kind of hangs together with what we know biologically. There's more such examples. This is another set of T cells. These are fibroblasts, which have roles in maintaining your extracellular matrix, endothelial cells, which are involved in tissue homeostasis. And the stories all make sense biologically. And so we got very excited by this and felt good about it for human data. We've gone on now and looked at other atlases we've made. We've looked at mouse, we've looked at lemur, and we're starting to compare them all to each other. And same story holds, and the biology is consistent across evolutionary distance, which we're super excited by. And we think this makes it a very promising approach to identify functions of cell types in a way that takes sort of human bias out of the question. And you'll see that that's a challenge in biology in other cases. In this next example, there'll be a particularly egregious example of human bias we think we've uncovered. So I want to change gears now and talk a bit about how we're using large language models to interpret the atlases. So the atlas is basically a giant matrix of numbers, right? There's a bunch of genes, there's a bunch of cells, and the matrix of numbers tells you the expression of each gene in each cell. And there's a question of how you'd assemble that into one sort of data object that you could interrogate and understand and study for relationships between the cells, and how you could do that across evolutionary distance. And in genomes, you assemble genomes like a jigsaw puzzle. That's a pretty clear assembly problem. It's much less obvious what to do in cell biology. So Yuri Leskovich and I decided a few years ago to try to build a large language model to do this. And we had two terrific students, Yusuf Rouhani and Yanni Rosen, who put this together. So the idea was to build a large language model on that giant matrix of numbers. Don't use any other information, no biology, none of that information going in. We didn't even want to use the genome. And so the basic idea is the same way that things like ChatGPT is built, okay, or Claude. They're built on text. You have a big neural network. You're trying to find weights that'll predict missing text. If you cover up a few words, you try to predict what's missing, and you optimize the weights to do that. And here we do the same thing, except we're covering up the gene expression. So in that giant matrix, we cover up 20% of the genes, try to predict them, and iterate the weights until we can do that as best as possible. Now, the other thing we decided to do here was make it work across all species. Now, normally, the genes are referenced to the genome of the organism. And so it's very hard to build tools like this that will work for multiple species. And Yuri had the terrific idea to use a protein language model to represent the genes. So these are models that are trained on protein sequences and create a sort of universal embedding space for any protein across all of evolution. And that embedding space is great because it's not tied to any single genome. And we can just take every single protein sequence for the genes or the genomes we're interested in, embed them here, and everything is on a sort of common footing. And so therefore nothing gets referenced to any particular genome. It's all based on protein sequence. And that ended up being very valuable. This was a sort of 650 million parameter model trained on about 30 million cells on 24 GPUs over the course of a month and a half. We had great ambitions for this to be sort of a zero shot, something that didn't need to be fine-tuned or retrained in specific tasks. And so we've tried to evaluate its zero shot behavior in several different contexts, one of which is to see how it handles data it's never seen before, that wasn't trained on. And so in the training, we withheld the unpublished tabula sapiens data. That was never part of the training set. It hadn't been published. And after the model was trained, we then embedded that data into the space that the model learned. And you can see the previous donors, the new donors, it all kind of lined up, and every cell went where it was supposed to. It had really learned what these cell types were and could do it with data it hadn't seen before from completely new donors with different genomes and different, they're humans, but different genetic backgrounds. Then we decided to test it on a creature it had never seen before, so a novel species. And this was another held out data set from the literature on the green monkey. And so it hadn't been trained on that. This is single cell data, another group published on lymph node cells. And we were able to embed it in the space, and we asked which human cells were nearest to these green monkey cells. And if you look and call that the predicted cell type, you can see the model did a pretty good job of assigning cell type function to cells from a species it had never seen before, which was terrific. Then we decided to start looking sort of more systematically at distances between cells in the space, and found that it really does represent biology in a way that was meaningful. So T cells, different types of T cells, we talked about a few of those in the last few slides, they're all pretty close together, and they're close to other immune cells. Whereas endothelial cells from different tissues, different types, they're all together in the same branches of the tree. Muscle is over there, and it kind of made sense. It had learned biology simply from that matrix of high dimensional numbers in a way that made sense. And a more general analysis of that, if you use sort of a formal cell ontology trees and ask, and sort of one cell begets another in lineage development, parent-child cell relationships have a certain distribution of distances, siblings have another, cousins a little further away, distant cousins even further, and so forth and so on. So on average, the further away in the embedding space, the further the relationship between the cell, the more distal the relationship between the cells. So that, sort of emergent behavior, we thought sort of wildly exceeded our expectations of how this would work. And then at CZI, we decided to build a second generation model where we trained a little more systematically across evolutionary history. And in the UCE, the first model, almost everything was human, a little bit of mouse, and a couple other species sprinkled in, and we didn't really systematically vary that. So when Theo Karalitsos joined CZI to lead the first AI group, he set about building a second generation model, which would be sort of trained more systematically and also have the ability to be generative, although I'm not going to talk about that today. You can read the paper if you're interested. And here we trained across about 1.5 billion years of evolutionary history in a way that was pretty systematic. And these are sort of the cartoon representation of the various creatures: human, mouse, rabbit, chicken, frog, fish, worm, fruit fly, single cell organisms like yeast and malaria. And I'll get into the reason why in a bit. And we didn't train just one model. Theo and his group trained a bunch of different models. So one model was on all those species, 100 million cells across the 12 species. And that's kind of the one that's using the most evolutionary information possible. Then we train a model only on human and the 4 major model organisms, which would be mouse, fish, fly, and worm. Then we train a model only on human and began to compare their performance, not just against each other, but against other such models in the literature. And one of the tasks we tested on was doing cell type classification. So I sort of gave the example of that previously with UCE. Here we did it more formally across a wide range of cells on data that was held out from the training again. And you can see that the F1, which is a sort of metric, is highest for our models, the 3 transcriptomic models and the UCE. And other models in literature are doing almost as well, but not quite. This one was not doing well. This is an important one here. This is sort of the control, which is the model that is not using any training on the cell data. So it's not machine learning. It's basically just using the ESM2 embeddings, the protein language model embeddings for each gene in each cell. So that's kind of like the control. And so doing all this training adds a little bit of performance, not as much as you might hope in this task. So we then decided to dig in and ask what's going on, which cell types the model is sort of saturated performance. Where is it strong, weak? So here, each column is a different model. Each row is a different cell type. And you can see for most cell types in human, all the models are doing equivalently well. But down here, there's some cell types where the models aren't performing equally well. And this little section is blown up right here, so perhaps you can read it. Here's the models, here's the cell types, and you can see that the transcriptomic models are doing the best. UCE is next, and the other models are falling off pretty badly. And the cell types here are mostly T cells. Those are the ones that are the most challenging to annotate and identify. And for those of you who are maybe biologists in the audience, there's been, it's not surprising, because for decades, the way to get published as an immunologist, the way to get your PhD as an immunologist, the way to get tenure as an immunologist was to discover a new kind of T cell. So there was a huge incentive to find ways to split and find distinctions that may or may not have been biological. And I think that's what's emerging here. The expert annotation and consensus, the ground truth is complicated. And it's that's what makes this problem hard. And this is sort of an example of where human bias and cell annotation creates real problems in how one understands this, the data. Other things we did were trying to classify cells as diseased or not. In this case, were they infected with COVID or not? There was data that was on human lung slices that Mark Krasnow and I published, single cell data that was withheld from the training. And then if you try to build a classifier to tell which cells are infected, which are not, again, the transcriptomic models are winning by a fair amount over other models. And so we hadn't taught the model anything about COVID per se, but it was able to recognize the transcriptomic differences. If you ask how well it does going across species, and how biology from one species can inform another. This is a data set also withheld from the training where the authors measured innate immune response to a perturbation lipopolysaccharide. That's a sugar that mimics bacterial infection. So it's sort of asking what's the immune response to infection in a simulated model system way. And they did this across a number of species. So they were looking across mouse, pig, rabbit, rat. And we would ask, well, if you measure in one, how well can you predict the response in the other? Again, sort of performance here, transcriptomic is doing the best. This is the control here, so substantially better than the control experiment. UCE is somewhere in between. So again, there's very interesting kind of emergent behavior. This is learning about perturbations in biology that it wasn't trained on. And similar results, by the way, for a cancer data set. As we start to think about evolutionary distance now, we can sort of really dig a little more deeply into this here. That was kind of one of the goals. So this is data from a number of primates besides human, chimp, rhesus, and marmoset. The chimp, rhesus, marmoset were not part of the training data at all. This is data from the cortex. And you can see that when you cluster it all together, if you color it by species, you see a bunch of sort of lobes in these clusters. Each of the colors are different species. Whereas if you color it by cell type, these are the different cell types, the clusters basically represent different cell types. And so what the embedding space has done is it has found a way to cluster cells of similar type together. They're close together in the embedding space, even if you're from different species. And so this cluster here is all, I don't know if this is the astrocyte or the oligodendrocyte cluster, but it's one cell type, many different species close together. And so this is a way to really sort of visually see that the embedding space is doing things that are sensible and interpretable as far as evolutionary relationships go. And so you can do a similar sort of thing of asking what are the nearest cell types across almost any evolutionary distance you want. This is now between zebrafish and rabbit. And if you ask for different cell types of the zebrafish, what's the closest rabbit cell? They're all ones that make sense. And so it really does what one would hope and expect as far as capturing these evolutionary relationships of cell type. And this then puts us in a position to ask questions about maybe origins of multicellularity and origins of different cell types. That's why we had the model train also on yeast and malaria, single cell organisms. And if you embed them back in the cell type, back in the embedding space and say, what's the nearest cell type to a yeast? Turns out to be progenitor cells. So stem cells are the closest thing to yeast, and there's going to be an interesting story to work out around that. There's sort of a similar problem you can ask about where do neurons come from. worms and frogs have neurons, but sponges do not. And there's been literature about potential neuronal precursor cells in the sponge. They're not neurons, but maybe they're the ones that gave rise. And the two candidates discussed are called choanocytes and neuroids. And if you take these all and embed them in the embedding space, and none of these were part of the training, you can ask, if you put the choanocyte in, what are the nearest worm or frog cells? They're both neurons, and they're both flagellar neurons that ciliated neurons that have some sort of sensory capacity. And the choanocytes are ciliated too, and so you can sort of see how these might be related. And these two could have been derived from this cell type. Whereas the neuroid, which was the other candidate, it's near two secretory cells that have nothing you can imagine doing with neurons. And this one, probably a false guess. And this is like a way to create test hypotheses that I think is going to be very impactful in evolutionary biology going forward. So we'll see, but where it's all going to go. But I'm very enthusiastic about the value of these tools and trying to understand relationships between cell types over health and disease, over evolutionary distance. It's going to be, I think, very powerful. Okay. And with that, I'm now going to turn towards another aspect of the problem that I sort of glossed over yesterday. I said pretty much every cell in your body has the same genome, except a couple, right? And the ones that don't are cells of the reproductive system, sperms and eggs, cells that have a few mutations picked up along cell division, and a few cells of the immune system. And I want to tell you a story now about these different genomes in your body. So now we're going to go to some of the genomic variation in your body. And again, to sort of set the stage a bit about sequencing DNA, and give you an idea of how much data there is out there, and therefore how much there is to interpret, let's put it that way. The E. coli genome is about 5 million base pairs long, which is about as long as the complete works of William Shakespeare. The human genome is about 3 billion base pairs long, which is about as many letters as in Wikipedia. To train ChatGPT was about 10 to the 12th letters, so more. But that's about 1/10 of the Library of Congress was used to train ChatGPT, an earlier version. In this publicly available sequence read archive, there's 10 to the 17th base pairs of DNA that have been deposited. I mean, we really don't know what that DNA means. I mean, it's been a massive amount of money and effort sequencing them, and we generated roughly 10,000 Libraries of Congress worth of data, and there's still many mysteries about how to interpret it. And thanks to Rob Phillips for these numbers. So E. coli, which is the hydrogen atom of biology. Let's talk about that first as a warm-up to looking at evolution in the human body, in the human race. Back in I think it was the 40s, Luria and Delbruck was a physicist who, by the way, if you've ever read Schrodinger's book, What is Life? He wrote this beautiful little book, What is Life? He talks about some of Delbruck's beautiful work trying to understand what the gene is. And there was the idea that it was a macromolecule. Delbruck did X-ray experiments to try to show that was the case. And Schrodinger interpreted that as the gene, this is before people knew what DNA did, the gene must be an aperiodic crystal, right? What a great description of DNA, an aperiodic crystal. I mean, he was definitely right about that. Delbruck went on to try to ask basic questions about how bacteria evolve. And he and Luria asked this question. This is what they won the Nobel Prize for. How do mutations arise? They come randomly over time, or are they induced by interaction with a challenging environment, like a virus trying to infect you? Either hypothesis could be true. They didn't know what the answer was, didn't know DNA was the molecule of inheritance, and yet they found a way to go after this. And there was this observation that bacteria don't necessarily need to be exposed to viruses in order to acquire resistance, and that kind of clues you into what the answer is. But they came at it from a very nice sort of statistical calculation they call the fluctuation test. And so in the case of induced mutation, that you don't see the mutations until you're exposed to the virus. You can imagine that a culture of bacteria that are growing up and then exposed to the, and then put on a plate and exposed to the virus, they will generate resistance to that virus at some common rate, governed by Poisson statistics. And that should be the distribution you see between replicates of the experiment. Whereas if resistance arises randomly in replication, you'd expect to see a much broader distribution of resistant colonies. Whereas, in this experiment, resistance didn't arise until the very last cell division. This one, it arose on the first cell division, and there's a huge difference in the number of colonies that result. So they did the math on that and did the experiment on that. And in the case of acquired immunity, induced immunity, Poisson variance over the mean should be 1, that's your Fano factor. Whereas in the case of spontaneous mutations, they calculated something much larger. Okay, depends on the mutation rate, number of bacteria, and a few other parameters. And they went and did that experiment and were able to show that it was not 1. It's a very simple thing. Just grow bacteria up in a bunch of test tubes, plate them out on Petri dishes, and count the number of colonies that grow. And this reveals something very powerful and insightful about the nature of mutation and the interaction of life with challenging environments. So we now have much more powerful techniques, and we know that DNA is the molecule of inheritance. We can sequence it, and so we can go well beyond what Lurie and Delbrück did. They used a model organism with a very well-chosen specific trait. We can now do this on a much larger scale in virtually any organism, we can't grow colonies from individual cells, but we can do single-cell genome sequencing to get the answer. And we started to do that in my group around a bunch of microbial problems. And this was the device we made to do that, where we could trap a half a dozen different bacteria and then lyse them, amplify their DNA, and get it off to sequence it. We used optical tweezers to manipulate the bugs. And this was, you heard the introduction, a little reference to biological dark matter. This is one of the great problems about diversity of life on Earth, that the vast majority of microbes, bacteria and such forth, can't be grown in laboratory culture. So the primary tool to study them is missing. You have to use indirect techniques. That's why it's called biological dark matter. And we were able to go and explore a bunch of these different systems, ranging from things found in the termite gut to things found in hot springs in Yellowstone, and all kinds of unusual organisms that weren't possible to get their genomes by any other means or their physiology. So having practiced on that device, we then built a bigger one that let us work with human cells, where we could amplify human genomes from single cells, and began to use that to study something about human genetic variation within one person. So there's a variety of ways in which we accumulate mutations and changes to the genome in our body. One is that we inherit them. The human genome is an average genome. We share 99.9% of our DNA sequences with each other, but 1 part per thousand is different just from inheritance, from familial differences and genetic diversity. There are mutations that arise during the course of our lives from environmental insults. If you smoke or are exposed to too much sunlight, you get mutations because the cells will make errors when they divide. Sometimes that leads to cancer. Sometimes there's errors just in copying the genome. The apparatus that copies the DNA when a cell divides is not perfect. It makes errors. Those accumulate over the course of our lives too. When we make babies, so making sperm and eggs, that mixes DNA from our mom and our dad, and also leads to mutations. And that's what I'm going to talk about. That'll be the one example I get into here. But just for completeness, finally, there's mutations that are intentionally generated. Your immune system to make antibodies is mixing and matching DNA. It cuts and pastes, it mutates it. That's how you create diversity in your immune system. We talked a little bit about that yesterday, actually, with the food allergy example. Okay, so let's talk about meiosis. So normal cell division, you've got your diploid. You have 2 copies of each chromosome. One you inherit from your mom, one you inherit from your dad. They're colored red and blue here. This is imaginary. There's only 2 chromosomes instead of 23. And normally cell divides, both are copied. Daughter cells both have a full complement. But in the germ cells, in the sperm and the egg, it happens a little differently. because if a sperm and an egg were both to be diploid and then come together, you'd have a tetroid. You'd have way too many genomes in the oocyte. And so in meiosis, the sperm and the egg divide, and they have what's called haploid, so one copy of the genome rather than two. And so therefore, when you meet your mate, you can contribute half your child's genome, your mate contributes the other half, right? Makes sense? High school biology? Okay. But there's mixing that happens in your body making these germ cells. So you don't just give a copy of your mom's genome or a copy of your dad's genome to your progeny. They get mixed together. And that's what happens through meiotic recombination. And so in this process, the chromosomes are sort of copied, they come together, and they can switch over different parts. And so the homologous chromosomes get next to each other, and they can have what's called crossing over, and we can see a variety of mixtures happen. And therefore, all the possible sperm or egg cells have unique genomes. Every single one is different through this random process of mixing. And your child will have some portion of the genome from their grandfather, some from the grandmother, and it's a random mixture crossed through. And we decided to study that at the level of one human being, one male, and the level of sperm. This was two very talented students, Jan Benoit and Christina Fan. And we wanted to look at this recombination potential for any given individual. There have been many studies of this process at the population-wide level. Things like HapMap, Decode Genetics, Molly Przeworski and Hutterite populations. And so people have been able to look at sort of averages across human populations, but nobody really knew in great detail at a genome-wide level how it worked in one person. There had been sort of point measurements, but not a genome-wide measurement. So we decided to do this with one human donor. Collecting sperm, it's not painful. There's a very easy way to do it. With that, put in the chip. The cells were lysed, the genome's amplified, the DNA recovered. DNA could then be genotyped and sequenced, and then mapped back to the reference, the average genome of the donor. Okay, and we had done something called haplotyping, so we had measured the continuous segments of the paternal and maternal inheritance of the donor's genome. And so for any given sperm cell, we could map back polymorphisms to one or the other. And by, sorry about that. And by doing that, we could work out for that given chromosome and that sperm cell, which were from the maternal part, which were from the paternal piece, okay? And there was crossover event right there. Does this make sense? Okay, so we did that on hundreds of sperm cells, and we're able to do this at pretty high resolution. This is sort of a technical slide. Each row is a different sperm cell. Each column is a different location in the genome. And if we were able to measure a single polymorphism there, it's colored in red or green. White means we couldn't measure it. That's the dropout rate. Blue is a case where we measured two, which is a problem. You only should see one. So there was either technical error or some mixing there. But in balance, almost everything is one allele, not two. And so we were able to amplify roughly 80% of the genome from each individual sperm cell. We then went and did higher density genotyping. This was like 48 different loci in the genome. This is like 500,000. The call rate goes down a little bit, but still 40% yield or so. And from that, we could do that mapping process I described for every cell that we sequenced, and we could look in great detail at what the recombination events were. So this is one single sperm cell. Each blue line is a different location in the genome where the genotype was measured and determined to be paternal or maternal. And you can see for each chromosome here, there's two columns. One is the maternal, one is the paternal. So it's called, I forget which is which. Let's say left is maternal. All this is maternal. And there's a crossover event here, paternal inherited there at that end of chromosome 1. Chromosome 2 started paternal, crossing over there to maternal, back to maternal, back to paternal, back to maternal, and so forth and so on. So we get statistics on all of these events and compare them to what was found in populations. And so if you look for any given chromosome at the cumulative distribution of recombination events, the lines are the population-wide measurements across thousands of people, many different families. The circles are the data measured from one single human being from hundreds of sperm cells. And they pretty much line up. It's kind of amazing. And you can look at this across all the chromosomes. sort of like an ergodic principle recombination. You can measure recombination events in one individual or across the whole population, and you're getting more or less the same answer at low resolution. Okay. Now, it turns out this recombination is not random. There's hotspots. There's parts of the genome that recombine preferentially. And that had been known. And we were able to see that phenomenon in our data. And there's a very interesting way. The statistical test for this is the birthday problem. You guys know the birthday problem. Turns out the generalized birthday problem doesn't have an analytic solution, but there's good approximations to it. But we found new hotspots that hadn't been found in the catalog before. And so it had all been averaged over by looking at broad human populations. And so this was a very interesting thing from the human genetics perspective. We then went and began sequencing. So genotyping is like low resolution sequencing. It's like looking at 500,000 spots in the genome. Here we just sequenced and mapped it all back to the reference genome. So the whole genome from a single sperm cell. And for every single, the DNA is cut up in little pieces. You sequence little fragments. You map it back to the genome. There's a degree or not, the genome. And that's what this histogram is, okay? For any given location in the genome, we ask, how well do the reads agree with what's there? And there's a giant spike here at complete agreement, okay? And this is like, if you can read this, 2 times 10 to the 8th. So roughly 3 billion base pairs in the human genome, all agree. Now, if you zoom in, there's a little blip right there. So let's zoom in. And this, instead of being a billion base pairs, let's make it be a couple hundred base pairs. You can see that not everything was agreeing. There is some discordance ratio at some of the spots. And this is due to things like sequencing errors, amplification errors. And they're minority contributors because mostly it's agreeing. But you'd expect this. I mean, this is just sort of the technical noise in the measurement. But then you get out to complete disagreement with the reference genome. And lo and behold, there's a giant spike here of a few hundred spots in the genome where every single read disagreed with the genome. What's going on there? We spent a bunch of time thinking about that, trying to find other sources of error in the measurement. Couldn't find any other sources of error. And so we said, we believe those are novel mutations in those sperm cells that came about through the chromosome replication process, the cell division, errors in the polymerase, and we think those are real signal. And we went and looked at several other sperm, and they all had a spike there at about the same height. And you can infer from this what the mutation rate would be. It's a little higher. So there was a sort of a family bulk rate in the literature, about 10 to the minus 8. We were seeing about 3 times 10 to the minus 8. But again, age matters in these things. Mutation rate should be higher in middle-aged men. So it seemed plausible. And we went and published that without sort of really knowing. But unbeknownst to us, the Decode group in Iceland had been working on a very similar sort of question from the perspective of family genetics and bulk sequencing. So they sequenced parents and the children, looked at the difference in the bulk genome between parents and the children, made a similar inference. And their paper came out after ours. This is their data. This is their figure. Age of the father at the conception of the child, number of de novo mutations. And this was our data, right on their curve. It was amazing. So happy that it worked out that way. With that, I hope I've given you sort of a taste of sort of the fun of and challenges in this intersection of biology, cell biology, and genetics and genomics. I'll just summarize. So, we started talking about this transcriptomes, so the RNA is expressed from the genome, and looking at what happens as we age in senescence. We looked at ways to identify cell type function in a principled way. We looked at the use of large language models to measure evolutionary distance between cell types across species. There's many other sort of things we're doing now. There's more for a biological audience. There's non-coding RNA. There's localization of RNA in different parts of the cell. We're sort of deep into that in the group now, but I think probably not as interesting for this audience. And finally, I hope you enjoyed the story about how genomes can vary within your own body. Thank you. I'm happy to take any questions.
With this like failed after that such that
So your question, I'm repeating it for the folks online if I got it right, is this question of evolutionary relationships. Does it capture spatial relationships between cell types and niches in the tissue? It's a good question. We haven't tried it yet. I would be very surprised if that happened. It would be an interesting result, not intuitive to me. There are people now measuring spatial relationships using what's called spatial transcriptomics. And as those data sets come online and similar models are built, then my expectation would be that would emerge from that. Elaborate on the which? Spontaneous mutation.
Certain sports or other examples, it's simple.
So the question is to elaborate on spontaneous mutations and whether there's analogies with fluid mechanics, phase transitions, boiling, that sort of thing. You know, it's how to say. Let me think about that for a minute. People think there's a decent understanding of some of the biochemical processes that lead to these spontaneous mutations, like the replication machinery, that's been well studied, those error rates, the context in which those errors happen, because it's also not random. There's sort of clear preferences for certain kinds of errors that are based on the biochemistry of the enzyme. That's maybe the closest answer to your question I can give. Is both from repair and replication. There's kind of the knowing the mechanism of how that works leads to an understanding of distribution of those mutations and the types of mutations in kind of a coarse sense. You know, I'll also just say that in this model of multiple mutations leading to cancer and tumor cells, you know, that's one where there's sort of a disjunction between the population genetics views and the actual measurements in human tissues, in the sense that, you know, the sort of two-hit hypothesis, Dolan, whatever, which is supported epidemiologically, people then went and found what they thought were cancer driver mutations. Then when they go look in normal tissue of people, those driver mutations are distributed in a way that's not really consistent with the model of them driving tumors. So there's like a puzzle there about, you know, what really activates the progression to tumor. And it's not as simple as just getting two drivers hit. There's a mystery there to resolve. It's not reconciled in literature. I'm sorry? Self-assembly. Yeah. Yeah. I mean, there's assembly involved in these apparatuses that do the replication and the repair. And so for sure, there's a molecular self-assembly aspect to it there. Yeah. Yep. Yeah.
why for like if there's like long distance like structure or and I think that's what we said too, like self-attention grammatical structure. Is that something like that?
Yeah, so there's, as you point out, all kinds of interesting architectures to play with. And my colleague Chris Ray at Stanford has been working on these long attention ones that were used for building EVO, the genome models. I mean, we did this first because it's the most straightforward. It's not clear to me it's the final word on the thing. I think we've opened the door to these things, and there's just going to be a very fertile area to explore going forward. Yeah. Sense of Sorry, using which statistic? Linkage disequilibrium. Yeah. I mean, that's where it comes from, basically, right? I mean, this is what drives those statistics. Well, we want to measure it without making assumptions. And, you know, I didn't get into it, but the story about how we measured the haplotype donor genome, there's a very interesting LD aspect to that, and come up afterwards and we can talk about that. Yeah. Yeah. Any what? T cells. Well, we have, I wouldn't glorify by calling it a theory, but you know, we have this idea that those are the least well annotated because of kind of the perverse human incentives in characterizing the literature. Yeah. We don't use the genome, but you could, I think maybe what you're saying is if you embed random genes in the protein language space and mix it up. Yeah, that's an interesting question. I don't think we've done that, to be honest. We've done different ablation experiments along the way, but I don't think we've mixed and matched genes from different genomes yet. It might be fun to try. Yeah.
This one here you can talk about attempts to rejuvenate cells. We talked about 120 years, and then you know, as you know, there are companies out there that are trying autos. There are many different things. Have you tried your approach to try to classify those types of cells?
Yeah, I did talk about yesterday our work on parabiosis to try to reverse aging with shared blood. And yeah, it's a very interesting story behind that. It's all published, and come see me afterwards. Yeah, I can give you the quick summary. Yeah. More complicated than that. Yeah, it's not about mistakes in the genome necessarily. And you know, the biology around it is super fascinating because they express factors that drive, inflammation is connected to it, and they're expressing factors that disturb the environment, and it's a very complicated set of interactions. Even the gene that, you know, drives it has multiple splice isoforms, and there's a lot of complex biology behind it, for sure. Yeah.
Looks very vague. Sentence itself looks like another state.
Yeah.
Does that mean what good? Does that give anything to sort of take it away? If it was like degradation, and you see there's so many ways of degrading.
Yeah. Yeah, they don't apoptose, right? Yeah. So the senescence cuts two ways, right? I mean, it was considered a good thing from the perspective of these cells are sick, there's something wrong with them, and they just decided to stop dividing, so they're not going to contribute anymore. So that's a good thing, right? And hopefully prevents cancer, things like that. On the other hand, they're secreting all these factors that drive inflammation, which is a bad thing. And so it's got two sides to it. It's not unambiguously good or bad. Yeah. Yeah. So we do a bunch of different dimensions there and just look at which one has the statistical best fit. Yeah.
Talk here a few weeks ago, talking about secondary and slash function. But I'm wondering whether your evolution of cells connects to that.
Sorry, what was the topic they were talking about?
Well, the topic of the popular book, time second arrow.
Uh-huh. Okay, time zero. Yeah.
And function.
Yeah.
Of the cells. So I was just wondering whether that's the function.
Yeah. I mean, there's this whole part of biology of convergent evolution where it appears that biology has solved the same problem in same way in different species, which would be statistically very unlikely. And so there's notions that the constraint space of biology is rather tight. And, you know, it's all been rather hand wavy, I think. People have collected examples. The box camera eye is a famous one. But I think these tools are going to help get at that in a quantitative way and help address that one way or another.
Okay,
Yeah.
so I think this can be the last question and we can wrap
Okay,
it up.
take them down here.
Yeah.
Yep, I'll be around.
Yeah, so I think we can conclude the questions here. So please join me in thanking Steve once again.